myself in the front of the room. Huh. Get out of that. Okay. Got very quiet as soon as I. Yeah, it's streaming. It's going. Okay. Do you want? What's that? You like that picture? I got a ton of great pictures up here.
I look so serious eating that ice cream.
You are so serious. Hello, and welcome to Cheyne Sprinbett's thesis defense. Before I go into and let him have the floor, I'm just going to say a bit about Cheyne before I let him do that. So, number one, Cheyne came to us from California. He makes that known fairly often that he is from California. That's one of his identifying characteristics, I'd say. And also Hawaii. You also came from Hawaii. You also make that known fairly often as well. So both good traits. And it was really nice because when I first had Cheyne come to the lab, there's actually quite a few things we have in common, which is great. Like, number one, he enjoys board games, and so that's good. Most of the people that come to my lab, if they don't already enjoy playing board games, they learn to love it at some point, or at least learn to tolerate it. So he's at least learned that. And he's also had an interest preceding coming to my lab in science communication, which is really good as well. Like, both of us really enjoy the art and the practice of communicating science well. So those were some things that really united us. There are some things that he learned at least to tolerate, if not maybe kind of enjoy, during the time that he was in my lab as well. It's really nice to see growth in students as they come into the lab and then they begin to learn new skills and new things as well. So one of the things is something like this. Like using more open source electronics for accomplishing research was something that he picked up along the way. And I think it was equal parts frustration for him, but also he, I believe, ended up enjoying it along the way as well. There's a few other things that were differences that at least I hope he's learned to tolerate over time, and maybe I've rubbed off on him a little bit in the wrong way. He was is still a very organized person and a very prompt person. That's also not two characteristics I share. And I know this caused him frustration more than once as he's been going through the lab, but he's always a good sport about it. And I think there's been a time or two it's actually rubbed off on him, and he's become somewhat more of a messy person, a little bit more fly by the seat of your pants person, I would say. But nevertheless, he's come to the lab, he's done really great work. He's presented this research already to good reception at SICB in Austin, Texas, where we had a lot of fun, but ultimately I've really enjoyed having him in the lab, and he's grown a lot while he's been here, and I've really enjoyed working with him. So with that, I'm going to give the time over to him to tell you about his research. Okay, I'll give you this as well.
All right.
Okay.
You guys can talk amongst yourselves while I'm setting this thing up. Hello, everyone. Thank you for coming. I'm really excited to be here to be able to share some of my research with you. I will just go ahead and get started. For thousands of years, sailors have fear of what lurks in the deepest corners of our oceans. And this fear has inspired countless stories and legends of monsters rising from the deep to bring chaos and destruction to mankind. Today, people seem adequately terrified just stepping out their front doors. And as a result, some of these classic monsters of old have become somewhat more manageable in recent times. But one thing has not changed, and that is the ocean is still a mystery. We are continuing to discover and learn new things about marine environments every single day, and only 5% of the ocean has been explored so far. Today, I want to share a story of another modern monster. This horrible little octopus will not be sinking any ships or taking down any cities, but it's still an animal that has risen from some of the deepest parts of the ocean to interact with humans. This is Muusoctopus leioderma, the smooth-skinned octopus, and it is a historically deep-water species. This octopus was discovered in a shallow-water marine sediment environment less than 10 years ago, and in that time, it has shown to be a remarkably fascinating and unique organism, even for an octopus. And that's saying a lot because octopuses are really cool. This is a charismatic group of animals with a high amount of public interest and relatively little research focus. Octopuses are highly intelligent, with some of the largest brain-to-body mass ratios of any invertebrates. On top of that, octopuses are ecologically significant. They are both generalist predators and prey, meaning they eat a lot of stuff, and a lot of stuff eats them. On top of that, octopuses are very widespread. You can find an octopus in every ocean on Earth. And that significance is expanding. Octopuses are increasing in both population numbers and geographic range. So we're finding more octopuses in more places than ever before. So again, this is Muusoctopus leioderma. That is typically found in around 650 meters of water. But for some reason, we have a population in less than 10 meters of water in the marine sediment environment of Burrowing Bay. And this is really exciting because it gives us access to an octopus that would be out of reach for most researchers. A little more on this species. This octopus is found throughout the northern Pacific Ocean, from Siberia all the way down to California. And there are a few things that differentiate it from other octopuses around the Burrowing Bay and Puget Sound area. For one, this is a smaller octopus. It lacks an ink sac. It has this really characteristically smooth skin, so it lacks the kind of raised skin folds and papilla found in other octopuses. Finally, this octopus has this really distinctive trait known as a keel. This is a flap of skin that protrudes from the lateral margin of the mantle, so around the perimeter of the octopus here. And I'll have better pictures of what that looks like in subsequent slides, but for now, just know this is a fairly novel trait among octopuses. And you may be wondering, what's so unique about this octopus? Well, this octopus doesn't just live in deep waters, it actually lives below them. This is a burrowing octopus, and this is a really novel behavior among octopuses. And this kind of leads into the central question that I wanted to address with this project. I wanted to look into this burrowing idea. How does Muusoctopus leioderma burrow? What are some of the specifics of this behavior, and how might burrowing be impacting this marine sediment environment in Burrowing Bay? So backing up a little bit, I want to talk about burrowers. So burrowing is a really common type of behavior. You see it in all sorts of marine organisms, and usually you see it in marine sediment environments. So marine sediment, like soft sands and muds, these are really common types of habitats. Most of the ocean floor is covered in marine sediment, and because of this, it's one of the most dominant types of habitats on Earth. And whenever you get into marine sediments, you generally find burrowers. Burrowing refers to moving through this soft sediment to form semi-permanent structures. And we have a couple different types of burrowers. So here we have a Norway lobster. This is a male fiddler crab. Right here we have a polychaete worm, and this is a mantis shrimp spear. So a big diversity of life that exhibits this kind of behavior. On top of that, burrowing is really impactful. It has a strong impact on the environment. Burrowing is what is referred to as a form of ecosystem engineering, meaning that these animals are actively modifying their environment with this behavior. Burrowers tend to have a disproportionately strong amount of impact on their environment compared to other animals. And I'm kind of showing just an example here of what this can look like. So this is what happens when we introduce a burrowing shrimp called a thalanassodean to a marine sediment bed that did not have any burrowers before. So over on the left, these arrows are representing water flowing smoothly over the surface of the sediment bed. Then you have this thick black bar, and this is representing a stabilizing layer of microalgae and bacteria. Finally, you have these little circles. These are representing individual sediment granules that are stacked on top of each other in this really uniform distribution. Then on the other side of this dotted line, this is what happens when we introduce that burrowing shrimp. As you can see, that water is no longer flowing evenly over the surface of the sediment. Instead, it's kind of ricocheting off the top of that burrow. And that stabilizing layer of microalgae and bacteria has been completely stripped away. These sediment granules are no longer uniformly distributed, but instead they are much blockier and clunkier compared with before. And this kind of change can be beneficial for some organisms, like microbes, that benefit from this kind of nutrient cycling and bioturbation. But it can also be a problem for filter feeders and other animals that rely on an undisturbed sediment bed. But the key takeaway here is that burrowing has a really strong impact on an ecosystem. And so getting back to Muusoctopus, again, that central question is, how does Muusoctopus leioderma burrow? And we're going to address this with three main lines of inquiry. We're going to be looking at patterns, performance, and morphology. Patterns. Does this octopus's burrowing follow any specific patterns? Performance. How does this octopus's burrowing compare with other marine organisms? And finally, morphology. Is there any anatomical traits, anything different about these octopuses that might allow them to burrow? And I'm going to start with patterns, specifically looking at burrowing activity. So octopuses exhibit both nocturnal and diurnal activity patterns, and this depends on the species. Muusoctopus leioderma has been shown to be mainly a nocturnal species, but this is just based on the fact that we usually can only find them at night out in Burroughs Bay. What we don't know is whether this octopus follows any specific patterns of burrowing, and we have yet to test this in a laboratory setting. And the reason this information will be interesting is that it would give us an idea into the type of ecological interactions this organism might be having. Next, I want to look at performance. And when I talk about performance, I'm referring specifically to burrowing rates. How quickly is this organism burrowing? And there are different things that can impact the burrowing rate of an organism. Sediment is going to be a huge impact here. So sediment basically dictates the strategy that an organism uses to burrow. So in thicker, more dense sediments, an organism is going to have to use a very different strategy compared with softer, less condensed sediments. And so that is going to translate to differences in burrowing rates. Next, we have species. The way a worm burrows is going to be really different from how, say, a crab burrows. And those differences are going to lead to differences in burrowing performance as well. And so it's important to keep these kind of things in mind as we look at the burrowing rates of this octopus and compare it with other organisms. Next, morphology. So we talked about patterns, we talked about performance, and now we're getting into morphology. Is there anything different about this octopus in particular that might facilitate burrowing? And I mentioned earlier that a really distinctive trait of this octopus species is the keel. This is this flap of skin that protrudes from the lateral margin of the mantle. So you can see more strong examples in these other octopuses. This one of Cortiformis is kind of exaggerated and artistic, but it gives you an idea of what I'm talking about. It's this flap of skin that protrudes around the perimeter of this mantle. Now, what's interesting is that, one, this is a fairly novel trait in octopuses. You do not see it in a lot of species. But it is also found in several benthic octopus species that are known to burrow. A lot of burrowing octopuses have been found to have this specific trait, this keel. And so the idea is that maybe this trait is somehow connected to this burrowing behavior. However, this has not been explored in any detail, and there's yet to be an exhaustive study examining this relationship. And this leads into the main objectives of this thesis. Again, that central question is, how does Muusoctopus leioderma burrow? And we're going to be looking at it by examining performance, morphology, and patterns. So first off, with patterns, does this octopus's burrowing follow any specific temporal patterns? Next, looking at performance. How does this octopus's burrowing compare with other marine organisms? And how is this impacting things like sediment and species? And finally, morphology. I want to get into the details of this keel, if there's any evidence that this is in fact a trait associated with burrowing. And I'm going to start by looking at burrowing patterns and activity. So to start, we had to find some octopuses. This is a map of our research area. We were up in the northwest corner of Washington State in the Puget Sound, up in the San Juan Islands. We were based down at Rosario Beach Marine Lab, just a quick boat ride from Burroughs Bay, that shallow water marine sediment environment where we find that octopus. And so myself and some helpful scientific divers went down and looked for these octopuses. We collected them and we brought them back to Rosario. Once we had these octopuses, I wanted to make a really comfortable, cozy environment for these octopuses to be held and essentially minimize the amount of stress these animals were experiencing while maximizing the potential that they would burrow in the lab. And there's a couple different components here. The first thing you'll notice is that when you walk into this room, it is kind of like stepping onto the red planet. It is completely 100% illuminated by red light 24 hours a day. And the reason we use red lights is that Muusoctopus leioderma, their eyes do not respond to red light. In other words, to these octopuses, this would be a pitch black room, and they would not be able to see this light source. And this is great because it allowed us to, one, walk around without tripping over anything, but also take pictures of this octopus at night, all without disturbing it with bright artificial lights. Next up, you'll notice we have these green LED arrays, and we have one of these on every tank. These lights were not on 24 hours a day. Instead, they were programmed to fade in and fade out following a diurnal cycle. So as the sun would rise, these lights would ramp up. They'd reach a peak brightness in the afternoon. Then they would slowly ramp down during sunset and turn off at night. And the goal here was to roughly approximate lighting conditions out in Burroughs Bay so that we could have a good chance of keeping them on their normal behavior patterns. You'll also notice we have this central water source feeding all of these tanks. Water was provided by the saltwater pump connected to Rosario Beach Marine Lab, and this was constantly pumping in fresh ocean water from Rosario Bay. And so this made sure that they were constantly getting cold, oxygenated seawater all the time, which obviously the octopuses appreciated. And each of these tanks was filled partway up with sediment. So we had 5 tanks in total, 1 for each octopus at a time, and these were halfway filled up with sediment that was also collected in Burroughs Bay. Finally, we have these little cameras here. And these cameras were really the main focus of this part of the project. So these cameras were programmed to take pictures whenever anything was moving in front of the lens. So whenever an octopus was out of its burrow on the surface of the sediment in their tanks, moving around, these cameras would take a picture. And those pictures would have file names with the times and dates that those pictures were taken. So this was giving us data on when these octopuses were active and out of their burrows. So we had all these pictures with this time and date information. And the goal here, again, is to look at burrowing patterns. So we use these pictures and this information to get an idea of burrowing activity. So octopuses were considered active when they were out of their burrows and moving on the surface of the sediment. So moving around, triggering those cameras to take pictures. Otherwise, octopuses that were motionless or down in their burrows for long periods of time, these were considered periods of inactivity. And so all the times when the pictures weren't being triggered were considered times of inactivity. In order to go through this, I had to go through and validate, along with a couple undergraduate helpers, that these pictures were actually being triggered by octopus movement and not by, say, someone walking into the lab or a snail crawling on the side of the tank or something like that. I'll let you see if you can spot where the octopus is in this picture. But once we confirmed that it was, in fact, an octopus in there and not something else triggering the picture, we copied and pasted that file name with that time and date information into our data sheet. while ignoring those pictures that were not triggered by octopus activity. And as a final note, not all of these octopuses were burrowing. We had some octopuses that were burrowing and some octopuses that were not burrowing in captivity. Why some of these octopuses were not burrowing in the lab, we don't totally know, but for those non-burrowers, periods of inactivity were when they were motionless for long periods of time, usually asleep in the corner of their tanks. And so we had all this information in this data sheet, and then we could plot it out. And this is how we decided to represent this activity data. So if you look at the top right picture here, we have this figure. And this is what is known as a circular time histogram. So you can think of it as a 24-hour clock. So up here at 0, this represents midnight. We have 6 a.m., noon, 6 p.m., and then back to midnight. So a big 24-hour clock. Periods shaded blue are nighttime hours, while periods shaded yellow are daytime hours. And then we have these red lines. These are showing the frequency of pictures taken, the amount of octopus activity during each of these time periods. So for A, we probably had the most amount of activity between 11 p.m. and midnight. Then for the second most amount of activity, probably around 10 p.m. to 11 p.m. So this was how we represented this data. Once we had this, we looked for a significant difference between the proportion of activity happening during the night versus during the day. When we did this, we got a statistically significant result, and what we found was that the majority of the activity was happening at night. So a lot of this octopus activity, when these octopuses are out of their burrows and moving around, was happening during the nighttime. And this was consistent with field observations and what we would expect to see. And this is cool because this is the first laboratory kind of set of methods that confirms this behavior, and it may give us an idea into some of these ecological interactions that are happening out in Burroughs Bay. Next up, I want to talk about performance, looking specifically at those burrowing rates. So now, instead of collecting octopuses and bringing them back into the lab and putting them in tanks, we wanted to record octopuses and get an idea of how quickly they were burrowing, so a measure of burrowing performance. And in order to do this, essentially we went looking for octopuses and videotape them burrowing out in the field. So this was kind of our setup. I had this 3D printed rig with a GoPro and a dive light. This was actually the second one. The first one I dropped off the boat and we lost it, and it was way better. But this is the one we ended up using at the end of the project that I got a picture of. And then on the bottom, we have this PVC waterproof housing that held our laser module, so this thing here. This was, again, a custom 3D printed design that I designed, which is why I broke like 50 of them. But it got the job done. And what this basically allowed us to do was whenever we were recording those octopuses, we ended up with these 2 red dots on the screen. And these lasers were a fixed distance apart and parallel oriented. So what that means is that I know the distance between these 2 dots whenever I'm taking a video is exactly 42 millimeters. Then I could take this video and run it through an image processing software called ImageJ and use this to set a scale. Then I can draw another line and essentially measure the length of the mantle of this octopus. So from this, I got both octopus lengths and videotapes of the octopuses burrowing out in the field. But the real goal is to get a standardized measurement of burrowing rates, a measurement of burrowing rates that we can compare with other organisms. This is the most commonly used equation to get a standardized measurement of burrowing rates. We need 2 things in order to calculate this. We need burrowing time, and we need weight of the octopus. So for burrowing time, this was fairly easy. We just looked through the video, marked when the octopus started burrowing, when the octopus ended burrowing, and that gave us burrowing time. Weight was a little more tricky, and the reason for this is once an octopus goes into its burrow, you cannot get it out again. Once it goes down into that hole, it is gone forever, and you are not seeing it again. So we could not collect those octopuses to bring them to the surface to weigh them. But what we did have was lengths, and we did have lengths and weights for another group of octopuses, specifically those octopuses that we collected and brought back to the lab. So for those octopuses, we measured lengths in the exact same way using those lasers and the GoPro, and then we also weighed those octopuses before transferring them into the tanks. So with this, we could create a relationship between lengths and weights of these captured octopuses. And once we had that relationship, we could calculate weights of octopuses based on those lengths that we had collected for these ones. So that's how we essentially turned lengths into weights. So we had our weights, we had our burrowing times, and now we had a standardized average burrowing rate index, or BRI, for this species that we could compare with other organisms. So this is a recording of Muusoctopus leioderma burrowing out in Burrows Bay. So I want you to kind of watch this octopus burrow and think about the kind of performance type of things that are happening here. Watch the strategy this octopus is using to burrow. Basically, it sticks its arms down into the sediment. It kind of gets acquainted. Eventually, it forces its arms all the way down, dives in after its arms, and eventually gets all the way down into its burrow. This is actually a slower burrower compared with other ones that we recorded. And you can see that keel pretty prominently right there, that line of skin. Okay, so keep those ideas in mind as you watch this video of another burrowing organism. So this is Octopus carna. It's another burrowing octopus that utilizes a radically different method for burrowing. So this is Octopus carna, like I said, another burrowing octopus, and it actually has a really cool strategy for burrowing. Instead of kind of forcing its arms down and pushing itself through the sediment, it burrows completely hydraulically. So it fills it up with water, and then it blasts that water out of its siphon, fluidizing the sediment and basically making that sand act as a fluid that it can then burrow into. So this is pretty cool, very different strategy from Muusoctopus. And there's a few similarities between these two species. One, they're similarly sized, and they're similarly sized octopuses, and they live in similar water temperatures. Although Octopus carna does live in slightly warmer water temperatures. However, the big difference between these two is sediment. Octopus carna lives in a larger type of sediment that is more sandy. So this is a coarser, larger sediment granule size. Muusoctopus live in Burrows Bay, which is a much finer, siltier mud marine sediment. And so these two offered an interesting comparison. But I wanted to go a little further, not just compare these two octopuses, but compare burrowing rates for a bunch of different marine taxa. This would allow us to kind of put that into context. So I pulled tons of burrowing rate indices because, as I said before, this exact strategy has been used in a lot of different organisms. And I pulled burrowing rates for all these different types of burrowers. So I'll kind of walk you through this figure here. We have sediment granule size in micrometers on the x-axis. So over on this side, we have larger, more coarse sediments like sand and gravel. And then over on this side, we have much finer sediments like silt and mud. Then on the y-axis, we have a burrowing rate index. And these BRIs are actually logged. So we have 0.001, 0.01, 0.1, 110, 100. And this is just to make the plot a little easier to digest. Then we have three categories of organism, and that's the different colors that you're seeing. So we have bivalves, which are burrowing clams in different shades of blue and purple. We have crustaceans in different shades of red. And then we have octopuses in green. And each of these little dots is showing a measurement for a single organism in a different type of sediment. So for this one type of bivalve here, you can see it was measured in all these different types of sediment, and these are the burrowing rates associated with that type of sediment. So each of these connected lines is showing an individual animal, different burrower. And one of the first things you'll notice is that we have a lot of variation with species. So each of these lines has a big amount of space between it. If you go up and down this y-axis, you'll see that the biggest difference between burrowing rates for all these different areas are species. Different species burrow at different rates. But we also kind of see this trend with sediment size. So here you can see what we get is this kind of like mountain peak sort of relationship, where you have a maximum ideal burrowing rate in this kind of intermediate sediment. And then as you move towards coarser and towards finer sediment granules, you kind of fall off the hill. And so your burrowing rates get lower and lower as you get away from that intermediate maximum burrowing rate. When we look at Octopus carna and Muusoctopus leioderma, we have 3 different measurements for Octopus carna, and we only have Muusoctopus leioderma measured in its native sediment. When you look at these two, the first thing you'll notice is that Muusoctopus and carna they're basically the same burrowing rates. Muusoctopus is faster than Octopus carna for this like really coarse type of sediment, but Octopus carna is faster for these 2 sediments. So there's not a huge amount of difference in burrowing rates between these 2 species. However, if you look at these 2 organisms, you'll notice this kind of nonlinear relationship, this mountain peak shape holds up between these 2 organisms as well. And so this is an interesting thing to see, this kind of U-shaped nonlinear relationship between sediment size and burrowing that may be holding up across other marine taxa. Finally, we want to tackle morphology, looking at the role of the keel and its possible connections with burrowing. So I mentioned earlier that one of the first clues into the idea that this was a trait associated with burrowing was the fact that this trait tends to pop up in a lot of different octopuses that are known to burrow. Again, when I'm talking about the keel, I'm talking about this line, this kind of flap of skin that protrudes from the lateral margin of the mantle. And so this first clue is that we see this in a lot of different burrowing octopuses. And so I wanted to examine this relationship and try to find some phylogenetic evidence of this association, and specifically look for an evolutionary relationship between these 2 phylogenies. But we also had a second clue, and this has to do with Muusoctopus leioderma specifically. So when we had this octopus in captivity, we noticed that some octopuses burrow and some octopuses do not burrow. But we also noticed that for some octopuses, that keel, that flap of skin, will actually get smaller over time. So as we hold this octopus in captivity, that keel will get less and less prominent, and eventually it will disappear altogether. And so the first question is, are these non-burrowing octopuses, the octopuses who are not burrowing in captivity, are they losing their keel more quickly compared with the burrowing octopuses? And if that was the case, the next question would be, does that mean that reduced burrowing activity in captive octopuses is leading to the loss of this keel? And if this were the case, this would be more evidence in support of the idea that this were a trait associated with burrowing. So first, I decided to tackle octopuses as a group. And in order to do this, we basically conducted a survey. So I went through and made this big, long list of octopuses focused on benthic species with genetic sequencing of these 3 genes, CO1, CO3, and 16S. And that was because these 3 genes gave us the best genetic coverage of the most number of octopuses. Once I had this list of octopuses, I asked 2 questions for each species on that list. 1, does this octopus have a keel? And 2, is this octopus known to burrow? Once I had this information, I pulled accession numbers from GenBank, I got genetic sequences, I aligned those sequences, and I created a phylogenetic tree. So this is a phylogenetic tree, and it's essentially showing the genetic relationships between all these different species of octopus. One thing you'll notice is we have these 2 colored rings around the perimeter of the octopus where all these different species names are coming out. The inner blue ring is showing the known keel. So species highlighted dark blue are species that are known to have a keel. The outer red ring is showing known burrowing behavior, with species highlighted dark red being ones that are known to burrow. Now, once I had this tree, I could test it for an evolutionary relationship between these 2 traits. And I did this with a statistical test known as Pagel's test. Pagel's looks for coevolution between 2 binary characteristics, 2 yes or no questions. Yes or no, does this octopus have a keel? Yes or no, is this octopus known to burrow? And when I ran this test, I got a statistically significant result. And what this test told us was that the model of best fit for this tree was one of interdependence. In other words, based on the structure of this tree, the most likely scenario is that these two traits evolved together. And this is obviously really exciting because this is the type of result you would expect to see if this were a trait associated with burrowing. Next, I wanted to narrow my focus and look at Muusoctopus leioderma specifically, and see if there's anything to this idea of non-burrowing octopuses losing their keels faster and more consistently compared with burrowing octopuses. And I did this by essentially going through those octopuses that we had in the tanks and taking periodic measurements of the keel, taking pictures of the keel as it was held in captivity over time. So once I had all these pictures of octopuses and their keels at various points throughout its time in our tanks, we evaluated them for prominence. We had 5 levels of prominence, with 0 being a keel completely absent and 4 being a very prominent keel. And we differentiated each of these levels with description telling the differences between each of them. And so myself and those same undergraduate volunteers went through and blindly and independently evaluated those keels for prominence. Once we had all these results, I ran an intra-class correlation coefficient on them, and this is basically a measurement of inter-observer error. And what I found was that our results were really consistent and that these methods were repeatable. So before I show you the results, I want to explain what we would expect to see if this were a trait associated with burrowing. Again, the prediction is that the keel is related to burrowing in some way. And some octopuses are burrowing and some octopuses are not. So here on the right, we have a figure of 4 possible scenarios. We have keel prominence on the y-axis. And so up here, we have very prominent keels. And down below, we have a keel that's completely gone. And then on the x-axis, we have days in captivity. And then we have these 4 different kind of scenarios with our data. For the top left, this is a significant effect of burrow. So you can see there's a big difference between non-burrowers and burrowers. Non-burrowers seem to have higher keel prominence values. However, neither of them are losing their keels over time. And so this trait is not being lost differently between these 2 groups. They're both maintaining their keels over time, and the slopes of those 2 lines are the same. They're parallel lines. Over on the right, we have a significant effect of day. So here, both groups are losing those keels over time, but those lines are basically on top of each other. So they have the exact same levels of keel prominence, and the slopes of the lines are the same. They are both losing those keels over time at the same rate. On the bottom left, we have both day and burrowing being significant factors. So you can see that non-burrowers seem to have stronger keels than burrowers, and they are both losing those keels over time. However, the slope of those lines is the same. Those 2 groups are losing their keels at the exact same rate. Finally, we have this bottom right scenario. And this is a significant interaction between the effect of day and burrowing, with day also being a significant variable. Here we have burrowers in blue maintaining their keels over time. However, if we look at non-burrowers, they're actually losing those keels over time. So this is a significant interaction between burrowing and day. In other words, the condition of being a burrower versus a non-burrower is impacting the rate of keel loss, the slope of that line. And if this were a trait associated with burrowing, this is the scenario we would expect to see. And this is what we ended up with. So again, we have keel prominence on the y-axis, and we have days in captivity on the x-axis. We have burrowers shown in blue, non-burrowers in red, and here you can see the different results. So each of these little individual shapes, that's showing an individual octopus. And you can kind of track how that octopus's keel has sort of changed over time in captivity. Then we have these thick lines, and these are the results of our linear mixed effects model regression analysis. These are basically showing the overall trends we see between burrowers and non-burrowers. And if you look at the results of this analysis, you can see that burrowing octopuses are maintaining that keel over time, whereas non-burrowing octopuses are losing that keel over time. So the slopes of these two lines are significantly different. And again, this is the kind of result, this is the significant interaction between burrowing and day that we would expect to see if this were a trait associated with burrowing. All right, just to wrap up here, again, our central question was, does Muusoctopus, the question we set out to answer was, how does Muusoctopus leioderma burrow? And we looked at that using three main lines of inquiry: patterns, performance, and morphology. For patterns, we found that we found a pattern of nocturnal activity for these octopuses. So we saw this was stronger with burrowing octopuses versus non-burrowing octopuses. And, And this nocturnal pattern was consistent with what we expect to see in the field. This lines up with our field observations of this octopus. And this is really interesting because if you've ever dived out in Burroughs Bay, you'll notice that the diversity of life is a lot more apparent at nighttime versus daytime. When you're out at night, you see a lot more different animals versus during the daytime. And so these patterns of burrowing are really what you would expect to see and are lining up with these patterns of activity for all these different animals. So this octopus is interacting with a lot of these different animals during these peak activity hours. Next up, performance. We found that Muusoctopus leioderma and Octopus carna generally burrow at about the same rate. There's not a big difference between them. We did find that there was a difference between the effective species with different organisms burrowing at different rates, and we saw this kind of nonlinear relationship between the effective sediment size and burrowing rate index. Finally, we looked at morphology. We explored this idea of the keel and it being a trait associated with burrowing. And we did this with two completely independent sets of methods. And both of these methods gave us the results that we would expect to see if this were a trait associated with burrowing. Now, this alone does not confirm that this is a burrowing connected trait, but it tells us we're on the right path here. And the next question is, if this is a trait that is connected with burrowing behavior, what is the function of it? The keel is basically just a loose flap of skin flying in the breeze behind this octopus. It has no physical structure, and so it's really unlikely that it's being used directly to displace sediment. But I'm really excited to see what future grad students and future researchers do as they start to get into the nuts and bolts of exactly what's happening down below the surface of the sediment. The deepest parts of the ocean remain a mystery, but times are changing. Climate change, ocean acidification, and general human disruption are resulting in us finding a wide diversity of organisms in unexpected places. As a result, we might continue to see these deep water monsters rising from the depths to interact with our daily lives. But it is important that we remain vigilant for any clues into these organisms and environments that make up so much of the Earth's surface. And with that, I have a bunch of thank yous to heap out. First and foremost, thank you, Dr. Onthank, my advisor throughout this project. I want to thank Dr. Alan Verdi from Maine Maritime Academy, and my committee members, Dr. Brothers and Dr. Nestler, for helping with the project and helping so much with diving. Speaking of diving, I want to thank all the undergraduate scientific divers at Rosario this summer who went out with me to go look for octopuses, always in the middle of the night, usually in the cold, sometimes in wet wetsuits. And I was always the only one who didn't have to wake up early the next day. So I really appreciate all the help. I want to thank all those 3 undergraduates who helped comb through my data with me and who are continuing to help me keep our octopuses healthy and happy. We have 3 of them downstairs in the tanks, and I couldn't be doing it without them, so I really appreciate it. I want to thank all the octopus researchers who helped me out with basically answering questions about keels and burrowing on species that I didn't have a lot of familiarity with. Big thank you to the American Academy of Underwater Sciences for their financial support of this research, and thank you to Walla Walla University and Rosario Beach Marine Lab for hosting me for this master's. And with that, I will be happy to take any questions. Thank you very much. Ricky.
On the slide, we talked about your Keels test.
Pagel's test, yeah.
So you said there support for interdependence.
Right, so Pagel's compares 4 models: independence, interdependence, and then 2 one-way dependencies. So Keel dependent on Burrow and Burrow dependent on Keel. And the model of best fit that was based on AIC values was one of interdependence. So the simplest model that fit our data best.
Can you, I'm a little slow on what interdependence exactly.
So so there's 4 models, right? So you have independence, and that was that there's no association. And then you have one-way and two-way dependence. So the idea is that one is dependent on the other or the other way around. I don't know exactly how it calculates interdependence, but the idea is that both are pressuring each other. So where you see one, you see the other, and vice versa. If it was one way, if you saw a lot of Keels, then you would tend to see a lot of burrowing afterwards. So it's basically looking at the genetic structure and comparing pressures going either one or 2 directions.
So if I look at the colors, like you said, I wouldn't necessarily just with my brain
Right.
be able to see it, but the test.
Right.
Okay.
So I don't know exactly like the nuts and bolts of the statistics that are happening behind the test, but that's basically how it works. It's comparing those 4 models. Other questions? Yes.
Why are we seeing them in more shallow water?
That is a great question. I mean, the usual, I don't know for sure. I don't think anyone knows for sure. I can speculate. The usual suspect is climate change and warming temperatures. There could be a whole host of regions or reasons. I can't say confidently why exactly octopuses are ending up there. We do see them in deeper waters, like near that area, so like just off the sound, off the islands, in much deeper water. Still relatively shallow, but like closer to 200, 300 meters. We do see some Muusoctopus out there that they've caught in trawl nets, but I don't know exactly what they're doing in this really shallow area.
And did you say that we're seeing more octopuses?
So their octopuses as a group are increasing in range and population numbers, yes.
Why is that?
I think it's because octopuses are really well equipped to deal with a lot of the problems that are happening or a lot of the changes in the oceans. Octopuses are pretty resilient to things like ocean acidification. They do fairly well with warming temperatures. We've actually did another paper that I'm a co-author on where we looked at P-crit levels and like hypoxia tolerance in Muusoctopus leioderma, and we found that they generally do fairly well. They're able to acclimate to hypoxic conditions. So like, I guess in general, just saying octopuses are pretty hardy animals and they do well in a lot of different conditions, that generalist organism. Questions? Yes?
Thinking about the loss of the keel and kind of use it or lose it, do you see any other loss of function in these octopuses over the course of the experiment? Like slower response times, do they get more stressed or more depressed in the lab?
That's a good question. We weren't really measuring it. Part of the problem was it had to be a really hands-off experiment in order to kind of look at activity patterns. So we didn't want to be pulling them out of the tanks and disrupting them and measuring, like, doing specific measurements for things like response time or motor function or like cognitive behavior. Stress could definitely be a factor. I can't say for certain right now. But all we did was look specifically at those keel prominence values and how strong they were. Yes?
Did you see any like change in activity, like daily activity over days of activity? So like, you know how you had like the 24-hour?
Oh, right, yeah.
Yeah.
And I should probably go back because I realized when I was doing my conclusion, I kind of glossed over this. So we have 4 figures here, and I think I kind of skipped it. So we have burrowers specifically on this one for A, non-burrowers for B, burrowers entering their burrows for C, and then octopuses exiting their burrows here. So we saw a stronger nocturnal relationship with burrowing octopuses compared with non-burrowers. And I forgot to mention that, so I apologize. But we didn't look at daily activity. So we didn't compare time over days. But the cool thing about this data set is we could definitely do that. That data is in there. And I feel like we just kind of scratched the surface with all this data. There's tens of thousands of pictures that we went through. So I think there's a lot of interesting things we could do with it. Yes?
How did you know the octopuses weren't affected by the red light?
So their eyes don't respond to the red light. When you shine red lights into their eyes, they don't react.
So they didn't have a problem with like the lasers being pointed at them?
I mean, we tried not to shine the lasers in their eyes. I wasn't looking to see if their eyes were responding. My guess would be, yeah, lasers probably would hurt their eyes. Luckily, that was not a factor for that part of the experiment, but yeah. We know they don't respond to red light. I haven't shined a laser into their eyeballs yet, so I don't know if they would react. That seems kind of cruel. I wouldn't want to do that. But we kind of avoided shining lasers into their eyes, yeah. Other questions? Any online? No? Okay.
Okay, last chance.
How is the green light that you use mimicking like the sun accurately? Or like the light cycle that would be
Right. So we did that in two ways. One, we chose the ramp up and ramp down periods using like a sunrise, sunset chart. I mean, it was a digital chart. We used it based on an R script. So we chose July 31st, 2022 as our like set date for those ramp up maximum brightness hours and then ramp down periods. Because that was a date about halfway through our data collection period. And then, what was the second part of it? We also measured light activity. So we got basically like a baseline for the peak amount of brightness that they might experience down there. We used a light meter and dropped it down off the boat to measure light conditions down in Burroughs Bay. And then we used that as a baseline to set those maximum brightness levels. Any other questions? How much time we got? Okay. All right, well, thank you guys. I appreciate it. It's been a blast. All right.
He and his committee will have a nice little conversation.
Now's the fun part.